Hi, and welcome to Chapter 4. We're going to take a little tour of the cell, and this is Dr. Papke from Bio 101. Let's start out by thinking about the big picture. We're learning about cells, and cells are the fundamental unit of life. What this means is that we define life by whether or not something has cells. So we say like all organisms are made of cells. And organisms can be something as small as a bacteria, or it can be as large as an elephant, or even bigger. Cells are the simplest collection of matter that can be defined as being alive. And what that means is kind of like an analogy of how the atom is the smallest part of an element that can be that can be having those characteristics. Because atoms are not alive, but cells are. And so cells are the simplest collection that can be defined as being alive. All cells are related to all other cells by descent. So what this means is that cells arise from other cells, and you can trace that all the way back in time to the first cell. The last thing is that cells can actually be very different from each other. And so even though they can be very different from each other, they still have a lot in common. We're going to learn about some of the things that cells have in common and how they're different. We're going to learn a little bit about how we look at cells. And that's how we're going to start out, by looking at the idea of microscopy. That's like microscopes. And we're going to take a peek at some of the basic cell types in this first part of the lecture. So, one thing that we know is that microscopes help us see things that the naked eye cannot see. So like most cells, you can't see with your unaided eye. And you definitely can't see a lot of the details that you'd want to see with of a cell. So for example, right here, here's a cell, paramecium. You definitely could not see that with the naked eye. But interestingly, this paramecium is surrounded by a cloud almost of cilia. And in order to see the cilia, you really need to have a very good microscope. So that's what we mean by how important microscopy is to biology. So you've seen this kind of microscope. This is our basic light microscope. We've used these in labs already, and we use the symbols LM for light microscope. So something too small to see with the naked eye, but something that we can see in lab. So light microscopes work when visible light, like the sunlight, passes through a specimen and then through the lenses, and then that light is kind of bent and it's magnified, and it allows us to see things that we couldn't otherwise see. These are the objective lenses, and these are the ocular lenses. So it passes through, the light starts here, it passes up, and through this lens is bent and passes through this lens and then reaches your eye. There's 3 important parts to microscopy, a lot of which you've learned about in lab, but not all of these. So the first thing is magnification. And magnification is basically the ratio of how an object size is actually compared to the image size that comes through the microscope. So that's magnification. We talk about 10x or 20x or 100x. Those are the objective lenses. Remember that we multiply the objective lens times the ocular lens when we're trying to figure out total magnification. The next big concept is resolution. And resolution measures how clear the image is. It also helps us understand the minimum distance between 2 distinguishable points. What we mean by that is that if the 2 things are very, very close together, sometimes, especially in light microscopy, you can't tell that they're actually 2 different things. So that's what resolution is. A good example here is when we were looking at plant cells in lab, the cell wall is really close to the plasma membrane. They're so close that we just can't distinguish them. So the resolution of those light microscopes in lab is pretty poor, but they're still useful. The last important parameter here is contrast. And contrast is the difference in brightness between the light and the dark areas. And this is just a nice series of pictures showing contrast. This has very little contrast. These have a lot of contrast, but mostly because a dye was used on them, a red dye that's picked up differently by the cytoplasm and the nucleus. Here we have a nice contrast, and this is a different kind of contrast. So you can have different ways of doing contrast, and using dyes is one of those ways. Light microscopes can magnify pretty well, but the maximum is about 1,000 times of the actual spectrum, specimen. So what that means is your light microscopes in lab, if you multiply the ocular times the objective, 1,000 is the highest that we can go, 1,000 times the actual size. But there are lots of cool techniques that we can use to enhance this and allow like different cell components to be viewed, like stained with different dyes or labeled maybe with radioactivity. And we don't use radioactivity in lab, but still kind of cool to think about. A lot of the subcellular structures, those are the ones that are inside the cell, that include things like organelles, and we'll learn a lot more about organelles later. Basic definition here for you is that an organelle is a membrane enclosed compartment within the cell. Most of those organelles are really too small to be seen by light microscopy, although we definitely can see the nucleus under many circumstances. There's lots of different cell sizes, and this is a pretty good diagram of looking at different cell sizes. You can find this as figure 4.2 in your book, either your in-person book or your online book. And so just kind of looking at the scale here, we can see that the smallest bacteria are about 100 nanometers long, and that we can see mitochondria and nucleus. Here's the size of a human egg. Here's the size of a frog egg. Now you can see a frog egg with your naked eye. So we can see things definitely in the range of, smaller than a millimeter, but, not as small as a human egg. So, and then of course, human, like a whole grown human can be, pretty tall, several meters big. So, and actually, interestingly, some of the nerve cells in your body can be quite long as well. So we've talked about light microscope and how light microscopes help us see things in lab, but biologists are not limited to light microscopes when we're trying to see small things. We can use something called electron microscopes also to help us see really, really small subcellular structures. Those are the structures that are smaller than the size of a cell. Usually within a cell, sometimes outside the cell, subcellular structures. So there's two basic kinds of electron microscopes. The first is called the scanning electron microscope, or SEM. What an SEM does is takes a beam of electrons and scans across the surface of the cell, the surface of the specimen, whatever it is you're looking at. It shows you three-dimensional aspects of that cell. And you can see right here a very nice scanning electron microscope photograph of cilia. Again, these are just formed by bombarding the cell, in this case the outside of the cell, with electrons. Now then there's a transmitting electron microscope, which we see the same basic structures of cilia, but this time look at the picture here. We're not seeing the surface of the structure, we're actually seeing through the structure. We're kind of passing these electrons through the specimen. This allows us to see inside of things. So the transmission electron microscope allows us to study inside of cells, inside of structures. And scanning electron microscopes let us see outside of structures. We've also done some really interesting stuff with labeling cells. And what we see here are several examples of different ways that cells are labeled with different markers. What we see right here is a fluorescent marker, and fluorescent markers allow us to see interesting details, like the blue marker here does the nucleus, and these green markers here allow us to see chloroplasts, that kind of thing. We can also use things like confocal, which is a type of microscope that just gives you really sharp images of tissues and cells. New techniques and labeling have allowed us to see structures using microscopes as small as 10 to 20 micrometers. So that's pretty cool. All right, so now we can see cells, we can see structures of cells, right? And we're going to start thinking about the two basic cell types, really focusing on one, which is called the eukaryotic cell. And you can see that word right here, eukaryotic. Eukaryotic can be broken down like the EU, the U part of the word means true, and karyo refers to nucleus. So eukaryotic cells have a nucleus, and a nucleus is an internal compartment surrounded by membrane inside of the cell. On the other hand, we have prokaryotic cells, and you can see that word right here, prokaryotic cell, right there. And prokaryotic cell, pro means before, and karyo still means nucleus. So prokaryotic means before the nucleus. Prokaryotic cells evolved before the nucleus evolved. So they are simple cells without a nucleus. If we look at the prokaryotic cell type, there are two domains, and domain is a hierarchical categorization of cells. So there are two domains, bacteria, which most of us have heard of before, and archaea, which are a little less familiar with people because they don't cause diseases. So we just tend to not be quite as familiar with them because they don't hurt us. But they're still very cool and really important to the earth and our ecosystems. If we're looking at different types of eukaryotic cells, there's four. There's protists, also known as kingdom protista. There's fungi, which is known as kingdom fungi. Plants, kingdom plantae, and animals, kingdom animalia. All four of those fit under the cell type of eukaryotic cells, and they fit under the domain eukarya. So there's bacteria, archaea, each of those are their own domains, and then there's eukarya. When we in general, think about living things, we almost always think about the eukaryotic cell types because those are the ones that you can see, for the most part, with your naked eye. So let's do a really quick compare and contrast here between prokaryotic cells and eukaryotic cells. First of all cells have four things in common. Doesn't matter whether you're prokaryotic or eukaryotic. There's four things that there is in common. There's the plasma membrane, which you can see right here, the plasma membrane. And then there's this fluid inner filling called either the cytosol or the cytoplasm. We use those terms interchangeably, cytosol and cytoplasm. So there's no difference between them in my book. I know that there might be some subtle definitional differences, but in my class, we're just going to use the terms interchangeably. Plasma membrane and cell membrane, I also use interchangeably, so don't worry about it if I use one word or the other. So far, all cells have plasma membranes. They all have cytosol, also known as cytoplasm. They have chromosomes. Chromosomes are made of DNA, and they carry the genes. We'll learn a lot about genes in a later chapter, but for now, genes are what keeps you alive, basically. And then all cells have these structures called ribosomes. Ribosomes are the structures that are used to make proteins. So 4 things that both prokaryotic cells and eukaryotic cells have in common. If we start to contrast them a little bit, we can start out by thinking about that word eukaryotic and prokaryotic talks about the nucleus. So one main difference is that eukaryotic cells have DNA in their nucleus. And again, the nucleus is just an organelle that has a membrane around it. Interestingly, it has 2 membranes around it. We'll talk about that a little bit later in this chapter. And then the prokaryotic cells don't have a nucleus. Instead, anywhere that you find the DNA is called the nucleoid. So it's not bound by a membrane. It's just like, here's some DNA right here. I'm going to call that the nucleoid region. Prokaryotic cells do not have membrane-bound organelles, and eukaryotic cells do have membrane-bound organelles. If we start looking more closely at the DNA of prokaryotic cells and eukaryotic cells, we find that prokaryotic cells' DNA is found in a loop. Look at that. It's connected all the way around. It's a loop. Whereas eukaryotic cells have DNA, we say it's linear or not in a loop, right? It's not necessarily a line, but it's definitely not, it's definitely not in a loop. So let's take a closer look at our prokaryotic cells. There's a lot of structures here. I am going to go through them on this slide with you. Unlike when we get to the when we get to the eukaryotic cells, when I start talking about all these structures, I'm just going to go through them one by one. But our prokaryotic cells are pretty straightforward. They're fairly straightforward and simple. So I'm just going to discuss all the structures that you would find in a prokaryotic cell on this slide. It's going to take me a few minutes. Play along with me. Let's start out by thinking about the structure that divides the inside of a cell from an outside of the cell. And that is the plasma membrane, or remember we also call that the cell membrane. The plasma membrane or cell membrane separates the inside and outside of a cell. All cells have that plasma membrane, remember, from a couple slides ago. So if we start thinking about that plasma membrane, that it encloses the cytoplasm and it allows different things in and out. It's kind of like a gatekeeper. That's what the job of the plasma membrane is. Again, we'll get more details on that later. If we're looking inside the cell, we could also look at something called the ribosomes. Remember, all cells have ribosomes, and ribosomes make proteins. Again, we get a whole chapter on how ribosomes make proteins later in the semester. We could think about the DNA, and DNA is found in the nucleoid region of the cell, of a prokaryotic cell. It's just wherever the DNA happens to be, that's what we call the nucleoid region. So here, there's the nucleoid region, because that's where the DNA is. We can also talk about the cytoplasm or cytosol, but it's not labeled on this picture. So if all of that is inside of the cell, ribosomes, nucleoid, cytoplasm, what's outside of the cell? And there's a bunch of things that are outside of the cell, including the cell wall. Now, not all prokaryotic cells have a cell wall, but if they do, it gives it a rigid structure. This cell that we're looking at right here has a rod shape, or kind of looks a little bit like a tic-tac. Those ones definitely have a cell wall. It's keeping that rod shape in place. Round bacteria, round prokaryotes, they don't necessarily have cell walls. The next structure that's outside of the plasma membrane, and it's also outside of the cell wall, is something called the glycocalyx, which you see right here. The glycocalyx is this coating on the outside of a lot of prokaryotes, but not all of them, that have it's kind of like this slimy capsule or something along those lines. And that helps keep it protected from being eaten by predators. I know you're thinking, what's a predator on a bacteria? So larger cells eat bacteria. Think about your white blood cells. The next structure that we're going to talk about are the fimbriae. That's this word right here, fimbriae. Fimbriae are also known as pilus. They help the bacteria attach to surfaces. Now, we don't always see fimbriae in all prokaryotes, kind of like cell walls and glycocalyx, but we do see them in some of them. And you cannot see them on a transmitting electron microscope. So they're pretty small and delicate structures. The last one that we're going to look at is the flagella. And flagella do what flagella do, and that is that they provide locomotion for our cells. So this cell can literally swim around in place. If we compare the size of prokaryotic cells and eukaryotic cells, kind of like this picture right here, eukaryotic cells are generally much larger than prokaryotic cells. You know, you got like a 1 to 5 micrometer diameter for a bacteria, but typically eukaryotic cells are anywhere between 10 and 100 micrometers. So if you look really carefully right in this region here, you can see a small round bacteria or rod-shaped bacteria. There's even some spiral bacteria in there. Then we see some yeast. There's a human red blood cell. Look how much bigger the human red blood cell is compared to that bacterial cell. But that red blood cell is quite a bit smaller than a human skin cell. Sperm are kind of in the middle. They've got a small cell structure right here, but a pretty long flagellum. So that makes them sort of bigger. And then the blue one is an egg. Over on the right-hand side, you see a nerve cell. So various sizes, but again, generally speaking, we have the eukaryotic cells being quite a bit bigger than the prokaryotic cells. If we look at the plasma membrane, and I told you I'd talk to you about a little bit about that more, the plasma membrane allows some things like oxygen and nutrients, waste, carbon dioxide. It allows those things in and outside of the cell. It goes in and comes out, goes in and comes out. Generally, if it's a cell, you're going to have a cell's made up of phospholipids. Remember, we talked about phospholipids in the last chapter. But plasma membranes have a lot more to them than just phospholipids. I do want to review really quickly here with you the concept of the phospholipid and how it orients itself in the plasma membrane. But we've got like a whole chapter on this. So this is just going to be like a brief introduction here to you on the plasma membrane. So here's your phospholipids right here and here. And the gray region is called the head of the phospholipid, and the yellow are the tails of the phospholipid. The head is hydrophilic and the tails are hydrophobic. The hydrophobic regions interact with each other, and the hydrophilic regions interact with either the water outside of the cell or the cytosol inside of the cell. We also have proteins that are throughout the cell membrane, and we also have carbohydrates that are like attached to those proteins, or sometimes they're attached to the phospholipids themselves. One of the things that we learn about cells is that because all the metabolism, and metabolism is just a word for all the chemical reactions that happen in a cell, all the metabolism that has to happen inside that cell, as a cell gets bigger and bigger and bigger, the volume is it's really getting much, much, much bigger. The surface area isn't actually getting quite as big. So if we look at the total surface area of one block here that's 1 by 1 by 1, the surface area is 6 of those, but the volume, we'll call that 1. So the surface to ratio to volume is 6 to 1, or we say that's 6. As a cell gets bigger, the surface area, let's say the surface area is 150 units, then the volume becomes 125 units. And that makes the surface to volume ratio 1.2. And that's like saying your surface and your volume are just kind of out of whack. However, importantly, if you start dividing that big cell into smaller structures, think about these as the internal compartments of the cell. If you start dividing them up into smaller structures, your surface area gets even bigger, even though your volume stays the same. Your surface area gets bigger because each of these little things has its own surface. And that's the genius of having organelles. Organelles increase surface area even when volume is not increased. So the surface to volume ratio there is back to a nice round 6. We are going to take a very close look now at the eukaryotic cells. And remember that eukaryotic cells are a lot more complicated. I grabbed this image off the internet just to show you some of those complications. A lot of these structures are divided into compartments called organelles. Again, I've used that word quite a bit now. Hopefully you're getting the picture of what an organelle is. It's just a structure inside a eukaryotic cell that has its own function, and it's surrounded by membrane, very similar to the membrane that I've already talked to you about, the plasma membrane. So we have plasma in eukaryotic cells, we have plasma membrane both inside and on the outside of cells. This is what our typical animal cell is going to look like. I know these words are small. I'm going to go through these with you one by one. So I'm not going to really worry about it too much on this slide. I'm just showing you kind of a preview of how complicated eukaryotic cells are and what we're going to be learning about. So this is an animal cell, and this is a plant cell. Main differences here between plant cells and animal cells are that plant cells have cell walls, they have chloroplasts, and they have a region called a central vacuole. Animal cells, on the other hand, have something called centrioles that plant cells don't have. But other than that, they've got a lot of structures in common. So we'll be going through all of those structures in the next part of this lecture, part 2.